Hi everyone, I'm Dr. Janina Jeff, Staff Bioinformatics Scientist here at Illumina.
Hello, I'm Elli Papa-Emmanuel.
It's so lovely to meet you.
It's great to be here.
Tell me a little bit about what you do.
So I'm an assistant professor at the Computational Oncology Service at MSKCC. So that is a way to describe computational biologists who perform applied translational research in oncology. And at the same time, I'm the co-founder and CEO of Isabel, a company that sets out to bring cancer whole genome and transcriptome sequencing as a diagnostic test in cancer care.
Wow, that's amazing. So we have a lot of oncology here happening at the conference. We have a lot of conversations around oncology. And as you know, we just released and launched a new product. Tell me about some of your first experiences, with Illumina and Illumina technology in your work.
Well, so Illumina has been at the forefront of developing and deploying technologies that allow us to really analyze the genome and extract the data. And as a consequence, it has really shaped my career path to date. So as an example, as I was finishing university, bioinformatics didn't really exist as a discipline. The human reference genome was just unfolding. As I moved into my PhD, and in fact, a key determinant of my PhD choice was to use new emerging technologies and data that looked at the genome comprehensively. So at that time, Illumina was launching the first Infinium arrays. So I was an early user and adopter of genome-wide array genotyping for linkage studies and genome-wide association studies. As I moved through my postdoctoral studies was the time where next-generation sequencing was being deployed. And now, as a faculty member at MSKCC, we're really leveraging the array of genomic technologies to answer fundamental questions in cancer biology and treatment.
One of the questions I have for you is, do you remember the first time you analyzed a full sequenced genome?
Absolutely.
Okay, tell us about it.
I remember even like the power, and it was an emotional time when you're sitting. In fact, I was sitting in bed on my laptop, and I was able to look through a patient's genome. And in fact, one of the very first genomic studies that I did with next-generation sequencing data was really trying to identify genes that cause cancer. So I had a very limited cohort of 9 patients for which we deployed whole exome sequencing at the time. And it's late at night, and I'm trying to learn how to analyze the data. And I see this recurrent variant that no one has ever seen before in 6 out of the 9 patients.
Wow.
And that was a mutation targeting a gene in the spliceosome machinery. Okay. So not a gene that would readily be identified as a cancer gene.
Yeah.
So the power of being able to look at an entire patient's genome and to be able to really pinpoint and identify not only what causes that patient's disease, but also uncovering a whole new pathway that had never been implicated in cancer before is a moment that I will never forget. And this is a moment that we've experienced over and over again as we have the opportunity to really dive into this type of data set using these technologies to understand disease biology a bit better.
Wow, that's an amazing story, because I have never. I feel like everyone remembers that pivotal moment of like when some type of new technology or data has changed, but a lot of us researchers rarely get that close translational feeling, and in that experience, this is translating into a diagnosis and a new pathology, or is that the word I want to say? But yeah.
Absolutely. That moment in time and that finding has now translated to a targeted diagnostic. new therapeutics have been developed targeting that very mutation. A new disease classification for the WHO, with also a lot of clinical trials now targeting this disease subtype. So it really speaks to, and you said not everyone has the opportunity to be so close to translation, but it's really the body of work that has gone into the development of the technology, the deployment of that technology that really enables us to bridge these studies to the clinic.
Yeah. So that was a moment that kind of really changed the work that you do. At this conference, we've been talking about what there is to come. What are you looking forward to in the future? Are there any type of genetic technologies you think that will come in the next 10 to 15 years that'll really impact oncology research?
So before we go into what is the next technology, what I'm really excited about and something that I'm thinking about is how now we can take these tools out of the research setting and really translate them to diagnostic tools that can transform the way we diagnose and treat patients. And that's a problem we've been thinking a lot about with regards to whole genome and transcriptome sequencing. So I'm very excited to be able to bring these latest advances, both in technology and data analytics, as well as knowledge, all the knowledge we've been gathering around how the genome is organized and structured, to patient care. Now, with regards to your question around emerging technologies, of course, now being able to go from a bulk tumor to a single cell resolution and being able to layer multi-omic data will enable us to really understand how the diverse cell populations in a given patient's tumor dictate disease biology and diagnosis, but also how they respond to therapy. And this type of single cell technologies will be instrumental in helping us understand what we do once a tumor develops resistance, how we can monitor those emerging clones, and how we can more effectively target these clones down the clinical course of a patient's journey.
How do you see multiomics as being a part of this picture? And I guess what are the omics that you'll be studying right now and in the near future?
Yeah, so I kind of touched upon this in the previous answer, but very much into what I'm excited about is really understand, like one of the biggest problems in curing cancer is the fact that even within a patient's tumor, you're not trying to treat a uniform cell population. There are multiple cell subpopulations or clones, each with their own deregulated network. And even though we may efficiently be able to treat the one clone, the other clone may escape. So implementation of multi-omic data such as single-cell transcriptomics linked to single-cell genomics will allow us to do subclonal phenotyping to understand how those clones look at steady state, post-chemotherapy, post-targeted therapy, and by understanding their biology, enabling us to develop therapies that are specifically targeting those networks that define resistance. So that's something I'm excited about.
Wow, you're incredibly, just listening to you, I'm getting so excited. Oh. One last thing, kind of unrelated. What type of advice would you give to a young computational biologist? I'm thinking back in 2003 when you were getting your first array data. What type of advice would you have given to yourself then, or what type of advice would you give to a young budding scientist in computational biology?
Yeah, one thing that I've learned is that the foundation upon which we work on changes and evolves. And one way, kind of staying on top of emerging technologies, data science is a real enabler of performing cutting edge research, right? Because technology moves so fast. And if you're so inclined to like technology and like data, being an expert at the most innovative tools that as they're coming out enables you to really define the future playbook of how we can implement these technologies in answering questions around biology as well as diagnosis and therapy. And another thing that I would like to add is that this is a problem that really requires bringing together individuals of multiple disciplines.
Yes,
So
I agree.
Data science alone, if it's not answering the right questions, can only take you so far. So working within communities of oncologists and cancer biologists and pathologists and understanding what are the critical questions we need to address, and then be the agent that can broker the implementation of the right technology to the right question, to the right sample types, is really key in creating a network of change and innovation.
President Obama was kind of alluding to this idea of having all these different voices in the room with different perspectives, different experiences, different backgrounds. I totally agree with you. I just want to thank you so much for taking the time and sharing this interview with me and having this conversation. It's been great to hear about your work.
Thank you so much for the opportunity to be here with you today.
Yay.
I really enjoyed it.